Welcome to Perception, or Psychology 347. My name is Bill Greiser, and I teach neuroscience in the Department of Psychology at Portland State University. And I'm going to be co-teaching this class with Jeff Leake, who is my colleague in both psychology and also in university studies. And he's also a professional working artist and quite an extraordinary artist as well. So we are very excited to sort of explore perception, both from a neuroscience perspective, a biology perspective, but also from an arts perspective and sort of an arts integrated perspective. So we're going to be not only learning about, the specialized sensory neurons, right? And the networks in the brain that develop with sensory experience into areas that are capable of supporting our perceptual experience of aspects of both, the world around us and the world within us. But we'll also be looking at how artists have, sort of intuitively understood a lot of this, and sometimes very explicitly understood, quite a bit of this, and how they can direct our, perception, through some rather extraordinary works. You're also going to get an opportunity to make a lot of things, to get in there and do things, to build stuff, including, brain cells, like this, and also specialized sensory neurons that we're going to be learning about in some detail, throughout the class. Here, just as an example, is an extraordinary, rod cell. This is a specialized photoreceptor that's, in your retina at the back like this that we'll talk about later. And this was created by one of our former students, as part of, the project requirements for the class. You don't have to make your specialized sensory neuron in this class out of, metal, but he went above and beyond, and, this thing even lights up, right? So we're going to be learning a lot about these rather extraordinary cells and the networks that they're linked to, and the role of, different regions of the brain in different perceptual aspects of our experience, again, from both a neuroscience and an art perspective. Now, this course is organized by weeks. It's also a hybrid class. So hybrid means that we're going to meet in person, actually, on the Portland State University campus, every Monday. So check our website, check the campus website to figure out the precise classroom location. And we're going to be, presenting material and having discussions and maybe doing, some interesting sorts of experiments on these Monday in-person sessions, during this hybrid term. We'll also be meeting on Zoom, on Wednesdays, for more sort of classroom instruction, and then also on Fridays, which will largely be review classes for, what was gone over, throughout the week. Please be aware that, we have lots and lots of material available for you to explore, and gain information and, learn more about these topics on Canvas. So there is a, under each week, you'll have many links and materials that you can read, that you should read through. There's no required textbook for this class. So all the materials that we're going to be offering are Creative Commons, NIH research, stuff that was already invested in by the public and is available for, sort of free public consumption. So every week, I'd like you to please read, the materials that we post, watch the videos that we put together that go over, specific topics, introduce them, and also in many cases review them. You'll be posting to discussion forums each week. So your first post, each week is going to be due every week by late Thursday night. That means, by 11:59 p.m. on Thursday, and then you're going to respond to two fellow students, posts by late Sunday at 11:59 p.m. So make sure that you are paying attention, and each week, basically posting in those discussion forums on Thursday and Sunday. And, we have a syllabus, and the syllabus is really important for finding out, what is due every week, what quizzes are going to be available, What art projects are we engaged in, going forth on? So make sure that you're also looking at the syllabus. The syllabus is right under the introductory modules, and if there's a change in the syllabus, we'll make sure to update it. There are 4 quizzes that you will, take during this class, and these quizzes are going to be available for multi-day periods. And you're within that multi-day period, you're welcome to start it, at any time. But once you start it, you've got to complete it. You got to go through to the end. So we're going to again have in-person classroom meetings every Monday, an online Zoom class every Wednesday, and then a Zoom review online every Friday. Now, sensation and perception, right? Perception involves, the sort of routing, the identification, and interpretation, right, of energy and information in our environment. Both our external environment, right, things that are striking our eyes and ears, for example, and body, but also our internal environment. We've got a lot of specialized sensory, receptors, specialized sensory neurons that we call sensory afferents, kind of arrayed around the body. And, aspects of somato, or body sensation, are a very important component of how we perceive the world, and certainly how we perceive ourselves. Oftentimes, perception involves kind of an initial detection of stimuli by specialized sensory neurons that we call afferent neurons, again, like a cone cell here, or the rod cell that I showed, and then the relay of this information along neural pathways, right, back towards the central nervous system. So in this class, we're going to investigate the physiological mechanisms, the specialized sensory neurons, right, the afferents, that are found primarily in the peripheral nervous system, we'll learn, although it'll turn out for the photoreceptors, those are actually, localized now within what we call the central nervous system. The retina is now considered part of the CNS. But we're gonna basically, explore the afferents that underlie our ability to detect, specific categories of stimuli, including light or electromagnetic radiation, right? Sound, chemical taste, and aspects of somatosensation, including a very important aspect, our ability to detect damage, to our own bodies, our own tissues. That detection of tissue damage, we're going to learn, is known as nociception. And again, there will be a specialized sensory receptor and a specialized neuron or afferent neuron that will respond and transduce, convert the injury information or something to do with the injury, we'll learn, into a neuronal response, and then start sending them back into networks of neurons, that we find in the central nervous system that are essential for our conscious perception, for, in this case, pain, right? So we're going to explore the central nervous system CNS networks that are involved in conscious perceptual discrimination, interpretation, and also more complex cognitive responses. And we're going to look at how these critical and adaptive neural networks actually develop, and how they are, they require, early sensory experience. I mean, it's remarkable that if a child, has cataracts, for example, an infant has cataracts, something blocking the light input to these specialized photoreceptor cells, for example, during these early, what we now call sort of sensitive developmental periods, where there's a tremendous amount of plasticity, ability of change in these networks to respond to that, sensory experience, and wire up appropriately. If they don't have input during these sensitive developmental periods, those networks are going to get kind of fixed in ineffective ways. So the child will not learn, will not discover, not develop the ability to perceive, what they are actually seeing. So there can still be a detection event, but they won't have wired up, appropriately during development, these networks that will allow for perceptual experience. This is actually a really fascinating thing. Like we require, early life engagement, right, with a lot of these sensory systems, like sensory, these energies and stimuli in our environment, in order to properly, develop the networks that we're going to be exploring this term, that link detection to, perceptual experience of, sight, sound, taste, smell, vestibular, input, a whole host of different things. We're also going to look at how these networks that are developed early in life inevitably decay. There's going to be loss of specialized sensory receptors, that'll occur, across the lifespan, and how, at a certain point we may lose that link, basically between detection and conscious perception. And that can often have this kind of counterintuitive the effect of kind of freeing up, the brain, these areas that are largely, we're going to learn, on the cortical surface of the brain, these networks that are involved in perceptual experience, like conscious perceptual experience. We're going to learn that they're going to be kind of, freer without, being tethered to the, previously had been the sensory inputs, to sort of hallucinate, to generate all sorts of unique, sorts of conscious perceptual experiences. So we're going to be reading a pretty extraordinary book that we highly recommend you pick up by Oliver Sacks, which is called Hallucinations. There are details, again, about this on Canvas. And finally, we're going to be talking about how artists have approached, understood, and integrated aspects of sensory detection and perception into the often extraordinary works, right, that compel, move, inspire, and affect our understanding of ourselves and our world. Jeff will be taking us on a tour of a museum. Potentially we'll do a live tour this term. But if not, we will be going, to a whole host of virtual museums around the world to explore, with new understanding of, sensation and perception, the works that have been created. Now, I just want to remind everybody that there's lots of material available for you to know what's going on as the class is proceeding here on the Canvas website. So the syllabus, again, all the due dates, all the assignment dates, all the quiz dates, right, all the art project dates, everything is going to be found there. So please check the syllabus and check for updates of the syllabus. There's often also under the modules important course information. Please go through these submodules. You can find, how do I find our weekly Zoom sessions, right? Who are your instructors? Who are your course teaching assistants? How do I schedule office hours, right? What art supplies do I need for this class? What are the expectations for this course? And then, very importantly, where can I go, to get help and support? Communication is really important. Please reach out to us, and the university has a number of very important and very accessible, sorts of support services that are available that we'd like to make sure you're connected to should you require them. And finally, Portland State has a pretty innovative, I think, interdisciplinary neuroscience minor where we're kind of exploring the nature of who we are, how we perceive, how we behave, how we respond, and what we're made of and how that changes, with environmental experience, from a multitude of perspectives. So we have an interdisciplinary neuroscience minor. And if you're interested in exploring more about that, you're welcome to take a look. There are links also available, here on Canvas. And finally, you're going to hear a lot about this. We like to take what we're learning and exploring in this class and other classes, that we teach here out into the community, to K-12 classrooms, right? To correctional facilities, to houseless youth organizations, to rural communities. So there are a number of outreach opportunities that will be available, and it's a great way also to kind of connect live and in person, in safe, kinds of circumstances, all around the country, and get enthused about what we're learning about, in this class as well. So welcome. Thank you very much for taking perception. I look forward to the term.